Welcome back to my YouTube channels. Hello viewers. In this video we are going to discuss about the PCR that is well known to us. PCR stands for the polymerase chain reactions. It is a test like and the other test like ECG. It's also a test biological test. So let's start the videos. First of all we will discuss what is PCR. PCR polymerase chain reaction, as I told you earlier, tests are a fast, highly accurate way to diagnose certain infectious diseases and genetic changes. The test work by finding the DNA or RNA of a pathogen, disease causing organisms, or abnormal cells in a sample. Uh, Now a PCR is just simply a test which is used to diagnose certain diseases, what form of disease could be. So these tests work for the pathogens and the abnormal sense. Pathogens, we say that the organisms that cause disease. Next is the steps of PCR. There are some steps involved in the PCR polymerase chain reactions, in which denaturation, and the second one is annealing, and the third one is extension. In denaturation, this is done at 96 degrees centigrade temperature, and heat the reaction strongly to separate or denature the DNA strands. This provides single stranded templates for the next step. And the next step is the annealing, and this step in polymerase chain reactions occur in at 55 to 65 degrees centigrade temperature. In this step we have to cool a reaction so the primers can bind to their complementary sequences on the single stranded template DNA. And the third step in polymerase chain reactions are the extension. It is done at 72 degrees centigrade. Raise the reaction temperature so uh, the polymerase extends the primers, synthesizing new strands of DNA. So these are the some basic steps that are involved in the polymerase chain reaction in denaturation, annealing, and extension. Next is about the electrophoresis. The ingredients of a PCR reactions are the polymerase, primers, templates, nucleotides, DNA building blocks. The ingredients are assembled in along with cofactors needed by the enzymes and put through the cycles of heating and cooling that allow DNA to be synthesized. Now we have to use electrophoresis. Electrophoresis is used to visualize the results of the PCR. After doing the test of PCR, we have to do the gel electrophoresis so that we visualize the results of the PCR. The results of the PCR are usually visualized using gel electrophoresis. Gel electrophoresis is a technique in which fragments of the DNA are pulled through a gel matrix by an electric current, and it separates DNA fragments according to size, and a strand of DNA later is typically used so that the size of fragments in the PCR sample can be determined. So in this way we have seen in this video what is PCR polymerase chain reactions and what are the different steps involved in the PCR and how we can visualize the results of the PCR through electrophoresis.